Hey everyone, welcome to our AgBioData webinar. So I have the pleasure of introducing our two speakers today. So I'm going to start with Adam. Adam Wright is a technical manager at the Ontario Institute for Cancer Research, where he is instrumental in the Alliance for Genome Resources, or sorry, Alliance of Genome Resources and Reactome projects. Um, he works on developing and integrating genomic data resources to advance biological research. He has background in biology and engineering, and he contributes to major initiatives like Wormbase, the Plant Cell Atlas Working Group, and Reactome to Go. And he's also been actively involved here in AgBioData, as we will hear today with his activities in the genome and gene annotation nomenclature working group, and also the Research Data Alliance, where he promotes bioinformatics, data integration, and open science. So thank you for working with us, Adam. I'm glad you were able to contribute to this working group and present today. And then next up, Dr. David Molik will also be presenting jointly with Adam today. He's the former computational biologist for the Arthropod-Borne Animal Disease Research Unit of USDA-ARS, and he has a new job. He is the incoming department head at Kansas State University for the Hales Library Center for Scholarly Publishing. If I can say so, David, you've assured me that you'll continue working with AgBioData. And it's probably actually even a better fit now that we have both the biology and now the library connection. So it's actually you'll still be a really great asset, or perhaps an even better asset now that you have those that library connection as well. So congrats on your new appointment there. It sounds super exciting. Um, but he did other work before this. So he did his postdoc with the USDA ARS in tropical pest genetics and molecular biology in Hilo, Hawaii, and got his PhD with Mike Frender, a colleague of ours at Notre Dame University in the Department of Biology, and with the Navari Family Center for Digital Scholarship. So we know David, he's a fair advocate for fair data, and also is one of our main contributors of AgBioData to the Research Data Alliance. Also works in I5K, which I'm heavily involved in, which is for the insect genomics community, and BioISAC, which I think is a security, biological data security working group too, right? So both David and Adam have their fingers in many pots. So I'll leave it back over to you, where you're going to talk about the progress that you've made and the tools that you've made for genome and gene annotation nomenclature. Right. So thank you.
Thank you, Monica. That was a great introduction. Um, Hopefully everybody can see the presentation. Good.
Looks
So
good, yeah.
Adam and I are presenting today for the guidelines for gene and genome assembly nomenclature working group, and happy to present. But this is, I want to recognize that this was very much a team effort, and I want to call out Effie in particular for making sure this work got done and got out, especially. Um, but for a reason, and honestly, I should say that she should be presenting, but for reasons she wasn't able to, so I unfortunately just get Adam and I today. It's a shame, but we'll carry on. Um, and can I please? There we go. So, as mentioned, we've recently published the new guidelines for gene and genome assembly nomenclature. This was intended to be a set of guidelines more than a set of like straight standards or requirements, something that could be built upon by various groups, um, but definitely sort of a framework and a thinking space. And hey, could you at least do this kind of research? Uh, Sorry, could you at least do this kind of naming scheme for your genome assemblies and your gene assembly um, annotations? So, nomenclature is surprisingly important. Clear, consistent naming practices are definitely linked to the usability and reproducibility of gene assemblies and the use of annotations. You can think of this as, well, a clear name allows you to even find the data in the first place. If you have nothing but assembly names on a list, and no metadata, then you couldn't even search. You just have to go off of the name. So, in that way, we can say that a well-structured framework for nomenclature supports collaboration and data sharing and cross-referencing repositories and different tools. Um, and so, really, as we see it, the objective of GAAN and the GAAN framework is just to, I don't define an approach for genome assembly nomenclature. So, the genome assembly nomenclature working group started by surveying across the different members of AgBioData and others, trying to find, what we're trying to identify problems, find what would be wanted in a good nomenclature for genome assembly and genes. And some of the problems identified were the lack of consistency across research groups and databases. So Group A uses a whole different naming scheme than Group B. Difficulty in integrating data from different sources due to ambiguous naming. So this list of assemblies is sort of hard to understand, or this repository is kind of hard to understand because the naming it doesn't really tell you anything about what's actually in the data that's being represented in that file. And the impact on computational tools requiring predictable naming structures. So I might use a small story here. When I was in undergrad, little Dave, I had a professor named Peter Fox, who was part of the Titleist World Constellation. And he would go on and on about the importance of putting metadata into the names of data files just on your own computer so you can understand, uh, what's being used. Well, it turns out that when you do that in a consistent way, computation tools can be set up to more easily mine and utilize the data of those files in a system. So as we built the framework, we wanted to incorporate the species name, the sequencing group, some sort, and continues to be a sort of nebulous idea of this is colony or population or breed or strain or varietal, something that is, as we talk about it, it becomes clear that it's something that is more targeted than just the species, as well as a version, which includes a subversion and other critical metadata if needed. We show you some different examples of how we use that other critical metadata. This ensures compatibilities from the previous survey discussions that the gene nomenclature working group had with others, and it aligns with major repositories like the International Nucleotide Sequence Database Collection for seamless integration. And hopefully get into, or I'll reveal that already gene assemblies have been submitted under this framework into um, NCBI assemblies. So, naming components in review contain a species, the sequencing group, the colony, breed, strain, more specific version, including subversion, and optional metadata. And um, we ended up going with the to standardize species names, which are a contentious and often debated, especially depending on the taxonomic group researchers in question. But the Darwin Tree of Life ID for species assembly seems to be a great resource. Most species that we have so far come across are represented in this list of species names that they've come up with. We therefore recommend their system, followed by a period, followed by stock, breed, subspecies, colony, tribe, population. The smallest possible grouping that makes sense, uh, sometimes even down to like the location that is a population, which is, this is not a perfect nomenclature. I'll get into why this can get, and I'll show you an example of why that gets confusing, followed by an institutional organization that did the sequencing, and we prefer the one that ordered the sequencing rather than the one that the where the technician actually loaded the sequencer. This is kind of a conceptual sense, if a ARS unit or a laboratory at a university sends off for sequencing, we'd rather know the lab um, or the unit instead of seeing this like Nova gene all over the place. Um, then the version of the assembly. So if in the way we describe this in the paper is if there's like a major change, that sort of is the major version. But if, say, you went back and you changed assemblers and assembled it in a different way where it was still the same sequencing data, that would be a subversion change. And then a optional miscellaneous, which could include information like a haplotype, organelle, callbacks about previous versions could put here as well as if you wanted to change naming schemes, you could just add the entire, for a version or two, you could add the entire name of the last scheme that you were using to go in or to start utilizing the new scheme so that you have both at the same time for some period of time. We don't recommend doing that for very long. So this is what a assembly name would therefore look like. So the Darwin Tree of Life ID Chilocoides sonorensis. And this one has been submitted to NCBI assembly, followed by, and actually this is the name of a colony within, and this is where we recognize the limitations of this. KS is Kansas, which all is simultaneously the colony but could also be a location. So there are limits. Followed by the sequencing group, in this case, Abidrew, followed by the version, the subversion, and a miscellaneous. Uh, we also looked at gene names and entities within uh, the annotation of a genome assembly. And so here you would add the assembly name, perhaps losing the miscellaneous portion of the assembly, followed by the name for the annotation, the chromosome the entity is on, the entity, which could be a gene, transcript, or pangene, followed by a uniquely generated number for that entity, followed by the transcript ID. We'll get in, I believe we have some examples of that. So here we lay out some existing assemblies, followed by the new assembly ID, followed by the new gene model ID. So what we present. Here we see, you can't see my cursor. Can I annotate? Yeah, maybe.
I can see your cursor.
Good, 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 good. I also found I can annotate. So here we see the Darwin Tree of Life ID, followed um, by the assembly group, followed by the version. There should be a subgrouping here, but since these are adopted, we don't list it here. Here we do. ID, varietal, version. This is missing the sequencing assembly group, and this orange part being the addition of the miscellaneous group. If I could just get out of that and switch. Oh, how do I clear all drawings? I'm gonna after this slide, Adam's gonna take over. But lastly, for my portion, I want to talk about integration with existing standards. The GAAN framework aligns with repositories like INSDC, which is GenBank, and EMBL, EBI, BB, DDBJ. It's designed to support future data sharing and interoperability platforms like Darwin Tree of Life Identifiers and Vertebrate Breed Ontology. Adam will get into a little bit about how we plan to extend software tools that we have developed for verifying gene names. With that, I'm going to stop sharing and Adam's going to take over.
Thank you, David. So, okay. Yeah, so to go with this, we created a first version, release 1.0, of a tool that works with this naming. Mainly that works with generating the names and then also validating names to make sure that they do follow the specification. Right now, it does a reasonable amount of checks based on the structure, but there are lots of things that we have in mind, and we have to design it, make it so that it could be more strict. So say if for a particular project, one of the sections needs to be from sort of a restricted list or something like that, then it would be nice to be able to make it so that they can use the tool and validate their names for their database accordingly. I'm sure there's other things that we could extend this to, but this is a great start. So here's the tool itself. So it's within the AgBioData repository or organization and has its own repository. It's reasonably easy to install with Poetry, and if you want, you can use Docker, and then it's a command line tool where you can provide, say here you're creating an assembly, you provide the parts, and then it will create the name for you. So it's a fairly simple tool, but very effective at working with this, and can be extended. So yeah, once again, this somewhat repeats what David was saying, but this, from a software engineering standpoint, this definitely, for furthering this tool or for other purposes, having this standardized naming structure can really go a long way for, say, pipelines that people are using to make sure that they are using the correct assembly, know exactly and not mix things up, that sort of stuff. Also, when people are just taking the file names and putting it in, whether it's other databases, like that these files are being submitted to, or just within a spreadsheet locally, that sort of thing, that you'd be able to organize and reduce the chance of error and understand what your data is much more quickly. Yeah, so this encourages, like, it also goes along with the whole trend of going and creating more and more standards and having things be interoperable between different databases and have users used to particular ways that things are organized, whether that's naming structures or other, but like and tools being used. But yeah, I think that this can go a long way to us furthering these sorts of tools. So this is a good example type tool, meaningful string. And then, yeah, so based on it being somewhat like a specification that different groups can implement, then research between different projects can be done much more easily. And people going between the different data sets will find the data more valuable, easier to find what you're looking for, and know with a standard way of doing things that they are accessing what they expect to be accessing. So not having to do things in a unique way per data set, that sort of thing, or even within a data set. So yeah, we hopefully the group is hoping that this is the type of thing that people could use across these different projects and be able to help facilitate the making data more open and usable to people. So I'm not going to go through all of these different bullet points. People have seen these sorts of things before. These sorts of standards or specifications are really good for making it so that people can find your work and meet these sort of standards, and make it so that from more so the trust perspective, the repositories that are hosting things, it gives a actionable way of making things easier to use and easier to find and all that sort of stuff from a particular repository. From the user's side, yeah, the increased operability and stuff like that will help them, especially for people doing studies across different databases. From like a development perspective of creating a new database or something, it's always nice to have specifications that are available that are being used elsewhere so that you don't have to develop your own. It's hopefully reduces the amount of effort as well. So yeah, at this point we're looking at doing a similar, like with this talk, like we want to go and do workshops. We published it, but like uh, engage the community and try to get people working with this framework, suggestions for the tool and for future work with this. But yeah, we're just starting into that phase of the project, and yeah, with the tool itself, like I was saying, like looking up ontologies or different resources like that to make sure that the names follow a bit more restrictivist guidelines would be a really good path forward, I think. So right now, just make sure that each of the sections exist, but if we knew that they've, especially for a particular repository, they could specify what the different the potential values for each of the sections of the names, then we can make a validation tool and a tool for generating the names that make sure it's more likely in line with all of the different files are for sure following the exact specifications they want. And yeah, people can give us feedback in various ways. One of the ways is with the GitHub repository itself. You can leave tickets that we've already had ones that we respond to, and yeah, so from the development perspective there, also feel free to engage with our working group. Anyone from the working group, I'm sure, would be happy to answer questions. Yeah, so in summary, we provide a robust standardized approach for naming the genome assemblies and gene annotations. We have approached different groups, including the INSDC, to make sure that this is in line with how they're moving forward and what they are expecting. And so this specification is something that can be submitted to those repositories. And then we are hoping that as groups, especially as more and more of these groups apply this specification, that this will become more useful across the different data sets. People become used to seeing these names and knowing what the different sections are, how they work, that sort of thing. And yeah, so our call to action is have people to try out this specification on your own data set. So we do very much appreciate feedback, positive or negative feedback, and ways to extend this going forward. Thank you very much. Yeah, I'll stop sharing. Are there any questions?
Yes. Thank you, David and Adams. Great presentation. We have plenty of time for questions and a pretty decent sized group. So Tanner has his hand up. Go ahead, Tanner.
Okay, well, can you hear me? I'm using a new system. All right. So I see that the paper is already published. So I wish that you guys came before present and maybe get feedback then. So I think this is a great effort. But I think that those coming from the wheat perspective, it took us months to come up with our nomenclature, and that was a great effort by the greater community. So I'm not sure if we will be able to adopt this new standards. They can be internal standards for databases, but overall community will still be using our TRAS gene models, for example.
Any responses to that, Adam, David?
Yeah. I think the Genome Assembly Nomenclature Working Group realizes that you know, not every group will be able to adopt the in, the nomenclature. However, and there are efforts that a lot of people have spent time on, and for a research community make a lot of sense within the context of their research, like wheat, rosacea, I think also has its own nomenclature. Uh, there are many, many groups and communities of researchers around different taxonomic groups that do not and have not thought about the nomenclature of the organisms in which they research. For them, perhaps this is a great, a great example and a great framework for them to have those discussions. Um, Hopefully, nomenclatures move in this direction. But it's a reason why it's a guideline and not, a standard, and why I shy away from using um, the word standard. Uh, I hope that helps provide some context to the work, Tanner. I'd be glad to
I
have it.
understand where you're coming from, obviously. And but adoption by communities, we learn, is very important, and they try to go with the shortest gene model, right? Shortest assembly. So when you add other stuff, like people will think, why do I need this, right? We know what's necessary. So I think again, it's for internal IDs. Like if you're dealing with thousands of genomes, you can have correspondence tables, for example, that they're very, very useful.
Thank you.
Thank you.
Yeah. So this is Kapil. Can you hear me?
Yes.
Yeah, hi. So Tanner, with regards to your question, so I think one of the pushbacks also we had when we were submitting this paper manuscript was also like, how will this community, how will the community adopt, and like, because they have their own standard nomenclatures that, that have been going for years. But then, it is at some point like the repositories that are already existing, they enforce some of these standards, and like but like as David mentioned, this is just like guidelines. But then uh, the group was doing like a survey initially that we wanted to get feedback and use that as a starting point and develop these guidelines.
Okay, thanks.
I'll,
Effie.
I'll make one more comment. From a sort of a pure computational aspect, just simply a unique number would be sufficient to uniquely identify all of this stuff. In working directly with researchers, I find that there's this thing, they don't want really long identifiers, and that's quite understandable, but then they want to know what they're dealing with. And so it's sort of you can't really satisfy both. And again, this is a suggestion. And yes, it's pretty lengthy, and it may work from some communities and not work for others to have such a long identifier. I know that the legume people who deal with lots of different species have fairly long identifiers out of necessity. So I agree, short's great. Numbers would be completely unambiguous, but people want to be able to recognize what they're looking at.
Good point, Effie. Thanks. I have some thoughts there, but I'll hold them to myself for a bit. Rex, I think you had your hand up next.
Yeah, I think there's two issues here. I've always thought there's two issues here. One is what the community needs or wants, and the other is what databases need or want. It is not necessary to make the community use only one identifier. If the community wants to use an identifier, then let's give it to them. However, we can have in the database, column that says synonym. And the synonym could be the Darwin Tree of Life number or whatever. Whatever identifier is best suited for exchange of information. Because ultimately, that's what we're really talking about with a standardized nomenclature, is exchanging information across domains, discipline domains, really. And as long as it's that identifier's in the record somewhere, it'll serve its purpose. The users in the community don't have to know it's there if we don't want to show it to them. We could always show it to them, and they'll look at it and say, "Oh, that's nice, but I'm going to use my old term." But as long as machines can get at it, all we need is a service out there that says, "Give me your Darwin Tree of Life ID, and I'll give you back whatever it is that we need to give back." Just my 2 cents.
Thanks, Rex. That might be something that the upcoming natural language processing working group might want to think about too. Anyway, Adam, Effie, David, do you want to respond, or all clear there?
Yeah. Actually, I'd love to respond to that comment, and I think it's a great comment. And my personal feeling is that synonyms are probably the way to go. I think there's some question in loss of information between synonyms across systems. System A is using synonym. Man, I've got food on the mind. Synonym, B, where system 2 is using synonym A. There are ways to handle that with different names. Something that Adam and I have previously thought about in other projects. But I do really appreciate that comment.
Well, I would add to that is that if there is a system out there that becomes widely adopted, then it's incumbent upon the databases to stand on their own 2 feet and go ahead and work that into the system somehow. I mean, that's just the price of admission, at least the way I look at it.
Sort of a both response and add on to what Rex is saying. When you have, say, a genome assembly that's in common use to a community, it quickly acquires a shorthand. Like in the maize world, it's B73V5, that's our current reference. So we have that. So if somebody comes, the maize GDB, new to maize, has heard that, and they type it in, they should be able to get the record for the current reference genome for maize. So I would say that, we probably should have mentioned something. I don't know, maybe we did mention something about synonyms in the paper, use them. But with some caution, because you can get cases where the same synonym is used for 2 different assemblies. If it can happen, it will happen eventually.
All right. Thanks, Effie. Lenore, you've had your hand up for a while.
Thanks for this. It's really great, and I really appreciate the sort of the editing function of the Git repo, and sort of both the recommendation and the tool thing. It's a really nice combination. Sorry, my brain just sort of went off into, I think I might have forgotten it. Well, I guess one of my first thoughts was around this issue of like the institution and whether or not there's the space for using, I think it's like the RRIDs, the registered, or ROs, registered research organizations. Like, is there a PID system in place already for entities that could be useful, so you don't have to worry about running out of alphabet letters? Though it definitely makes it even less human readable. But I think that's sort of the bottom line is like, the human readability of all of these is kind of gone way down, and that's maybe not really the point. Like I can't see typing one of those things into a paper a million times, right? So, I think the shorthand is kind of necessary. But yeah, I don't know if you looked into the RROs.
I,
You know what I'm talking about?
yeah, no, ROIDs. That's a fascinating. I actually don't know. To the specificity that you would want in the nomenclature or like in the name itself, whether or not there is a listing of research groups that is, or a way to make IDs that are for just research groups that are short enough that, you could add them into the name without making it too long, but long enough that you still have a general idea of what that research group is. We have thought about adding, I should say that part of what's coming on the tool is that, DTOL ID lookups for the species name, right? So easy enough to go off to the master list, which exists on GitHub in the raw format, find the ID, check that the ID that you've created exists. And we've thought about adding features that connect into ontology and, at least on the tool side, ontologies for various things. Look up whether or not the entity that they, want to put in the gene name is in fact an entity that exists within, the gene ontology. Easy enough, right? These are just, does this exist within the context of such and such a schema? Entity, I don't know the answer about entity IDs, but I would be fascinated to follow up on that particular question in the context of, we're already starting to think about some of these things.
All right. Thanks, David. Next up, Sudash. Sorry, I'm probably getting your name wrong. Go ahead.
Yeah, yeah, I'm Sudhanshu. Yeah, I think some of the points have been mentioned about the IDs, both in the context of genome assemblies, annotations, and gene IDs. Like she mentioned, in the journal, we use a common name of the gene, and the first time we mention in the article, we give the gene ID number, and subsequently we don't have to mention those long IDs. Fine. So if we follow that pattern, in databases, we can create another ID called standardized ID for assembly annotations and gene ID. And that would be the standard that people can adapt. Okay, and that not necessarily is human readable. That's for computational purpose only, like this is purposed for. And then, like we have gene ID in the database and a name, which in some databases is used as a common name, then we can have all other synonyms. And standardized ID will be the one for computation, and name or gene ID or other things can be that's friendly to the community and for communication, those kind of purposes. Probably it's naive to say, oh, it's just another ID for databases have to maintain, and that might be helpful. That's what I wanted to convey.
Yeah, that's a good point, that this is intended for an identifier and less for a functional description. It's good. Lenore.
Sorry, this is actually more to just kind of make a point that, but when Adam and David were talking about like wanting to get more feedback and the value of, reaching out to the broader community to get feedback, like I do think, having comment periods is really kind of helpful. And I think this has been presented a number of times at like PAG and our community meetings. But this is sort of a way of saying like, things are still trying to figure out a few things, but we should be able to support through the AgBioData creativity extension some awards for travel for people who want to present on outputs from the working groups. So, keep yourselves sort of tuned in for that. Hopefully we'll have some announcement about that fairly soon. Obviously, there's like a little bit of uncertainty. But I did sort of want to put that out there because it is important to be able to get reach out to those groups that maybe don't really have their established nomenclature guidelines yet. So that was just me sort of saying like there's this thing on the horizon, hopefully.
Lenore, thanks. Doreen.
I wanted to follow up a little bit separate than some of the things Lenore was saying, and really to the question that Tanner brought up earlier. So I do believe that the working groups have been communicating when they've been working on manuscripts, when they are looking for feedback and things, and perhaps there is the concern that we are all oversaturated, and sometimes some of these things go by us. And while the reach out has been by the group, it just gets lost in the noise. So I'm just wondering, is as a group, when we ourselves might be looking for feedback for this, because I know for a fact that this group, and I'm not part of it, has asked for feedback multiple times and had challenges in achieving feedback. How this group might think about moving that forward. Like if in these monthly meetings, as an example, I'm just giving it as a throw out there could be a period of 15 minutes or 10 minutes when there is a specific need for the group, and maybe there could be an ask during this time when there are more people available and their focus versus, for instance, the things happening. Because I do recognize what Tanner said about getting feedback is important and having that open time. I do think that this group did due diligence, but I think it got lost in everything else is going on. So as we move forward, I think the group should think about how to focus attention when those asks are happening.
A good question for the whole group. I'm going to add on briefly to that before I open it up. One of the questions and thoughts I had was, you have a GitHub repo for the software. Is there a resource for the standard, or you're not calling a standard, for the recommendation or the guidelines that could be versioned and commented on? I'm thinking, analogously to the GFF3 spec that is in a GitHub repo that, we've commented on before, but where, in addition to the software, the standard itself could be commented on, and that discussion could be held in GitHub issues, in addition to whatever is amenable for this community to support people who can't show up to this webinar or other ones that you're holding and might want to comment in a different time zone or in a different fashion. Effie, go ahead.
It would be great if we could do something like that. One of the comments I got from the preprint was a request to add some information. And my response was, oops, sort of, I'll do what I can. But this is not the last word, because obviously this is we've put it out there. People will respond, and yes, I think there will be a 2.0. So it'd be great if we can figure out a way to just open up comments.
So I was thinking specifically about what Doreen just said, and Tanner too, like, are there in danger of sort of inserting yourself into these communities, but are there other, local groups? Like, does the wheat community meet where you could have presented on this with the wheat people and for the webinar that they do on a monthly basis? Or, I'm thinking David for I5K, to say, hey, let's all move at least to the genome assembly standard. I have some thoughts on the identifiers for the annotations too. So is that another way to let's get feedback? And I know that's more work for y'all and you are over committed too, but have you considered that?
You know, we did review, for example, the wheat paper. We spent a lot of time with that, the wheat nomenclature paper, which was very impressive. And I know that it took a very long time to produce. And although it may not show directly in what we came up with, it did influence some of our decisions. But it would have been good to talk with the group too. That's true. Although a lot of the group is in Australia. But Tanner could have
You
been
mean
the rep.
yeah, for wheat, actually, that's a very international group, and
Yeah.
they're not usually the computational biologists. They are usually geneticists, breeders. So that was the driving force. Talking about standards to in a wheat setting is usually sometimes like an eye rolled, right? Like we all know.
Yeah.
But I think that this is a very important effort, and especially as I said, I said internal, like a correspondences table for especially large scale projects. This is a very important work.
Yes.
But I don't think it's a either or. So it's not a new standard. There is a standard, community standard. I think that needs to take precedence. And then you have this identifiers that are corresponding to these that we can use. Um, so
Yeah, we had hoped
I'm
to also put a letter to the editor, and the number one thing was going to be consistent. You know, use your community standard. Don't invent something new. So I think that was really the number one message. And then if you don't have a community standard, here's an idea.
I'll just chime in so I haven't said anything for a bit. But I think, yeah, we should, as a group, try to figure out ways to more utilize the GitHub issues and make it so that it's more obvious what the specification is and the changes being made through the GitHub repo. This is definitely something that can be done going forward. The second part of the discussion, I think, is much harder of how do you get it so that the right people are aware of it, because not everyone's going to be just sitting there watching our issues go by and everything as we make the specification. So definitely developing lists of people to reach to and having people be aware that where they would look within the repo to see what we are intending on doing going forward is, yeah, definitely something we can do through GitHub as the technology solution. Most of this is more of a outside of the technology, but we definitely will try to do what we can that way.
I've been holding back, but since I'm the moderator, I guess maybe one more thing. Tanya had in the chat a comment on DTOL IDs versus NCBI taxon IDs and Effie. I guess the answer to that it's primarily readability. But there are also a lot of people who are using that already too. So perhaps that's part of it too, at least with new assemblies from that we know in I5K, you know, everyone's using DTOL IDs for their assembly names. Um, so from a practical perspective, so I recently had a use case for this because I have been avoiding generating identifiers for the i5K workspace and using the NCBI services for that instead. But I had a use case where I actually needed to generate them, and I thought, oh, I'll just go and try to use the nomenclature here. And so I had a few comments there. One, you know, I agree with sort of the length issue for some, you know, use cases. If you're able to number your chromosomes already with a well-established assembly, it's like 1, 2, 3, 4. That would make it shorter. But for a lot of mine, they were, you know, NC underscore da 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 da. So that contributes to the length. The other thing that I ran into is that I'm not sure it's quite there yet for actually, somebody to actually implement practically. So if it, I'm not sure if it's possible yet to pass a GFF file, for example, to the program where it would know, for this feature, here's the chromosome, and I'm going to calculate or have this enumerated ID and have the sequential numbers so they're, essentially have that unique number string at the end. So some sort of technical implementation details that might make it easier for people like me to adopt, because I ran into that and thought, I don't have time to write a script myself to actually, pass the GFF and have it establish that. I think for the genome name that it works, but I ran into that. So that might be something that the group can think about, sort of easier implementation. But I will say, it was super handy. I did end up creating identifiers that are similar to that with, the thought of I really wanted to have the assembly name in there and the annotation name in there. So it's clear to the user and to me, like it's from this assembly and this gene set, even if that identifier is found outside of that GFF file. So I did appreciate having these sort of guidelines that I then riffed on for this sort of more unofficial identifier generation. So trying to use it and I'm hopeful to see more coming out of this so that we can use it a bit more in the future, especially as we're getting more genomes that actually have named chromosomes, where that probably aren't in the GFF file. So there might be a limitation for that too. Anyway, just wanted to throw out there, and so I appreciated that, and it does have some use, and it'd be great if you guys can find continued time to keep on working on that. Sorry, that was a conversation stopper. Okay, any other comments on that? Or Doreen Kapil, I saw you unmute a bit earlier. Was there anything you wanted to bring up?
I mean, I'd just like to say, I think the suggestion with the GFF is pretty good. I think we initially had a discussion very long time back when the group was started. Then this came up. It's just like, how to maintain it and like, do it. But I think since now we have the components of the identifier recognized, so it's easier now we can implement this in a tool which can basically take the GFF and auto increment the identifiers. So it's something that, yeah, we can pursue as well.
I also want to follow up on the recommendation early on about maybe some of the infrastructure that AgBio has and being able to provide comments, as Ethy said, because things are a living document. Perhaps maybe we can think about, as papers come out for standards from this that are authored predominantly by this working group, there might want to be a location of where that is, and people can provide additional comments and things there, so that as it's moving forward, it can somewhat be maintained that location until a new update happens. I'm just thinking because this group will have multiple products of recommendations of best practices, and we know that best practices get updated even every 3 or 4 years. And having those comments, especially when they're coming from other databases with examples and why or why not, those actually become very useful to include later on as well. So whether it's a GitHub, I mean, if this paper, for instance, had been on bioRxiv, there would have been an open period where people could have commented. Maybe this project could think about, for the papers that are authored with it, to be able to have a place to have those comments to continue.
Good point. Lenore.
Yeah, I was just, sort of asking if anybody had, certain suggestions. And I was thinking also maybe reaching out to Peter, who I think is very good with the BRAPI and sort of, having the community space to work on things. So I know that, we do have plans to overhaul the AgBioData website to certainly make things more transparent. And I don't know if any of that functionality includes the ability to sort of have discussions on that. I mean, we definitely have like a Git repo for the project as a whole. So yeah, I think, wherever, I mean, and we have, Zenodo as a resource. I think, Effie, correct me if I'm wrong, you tried to put it on like bioRxiv or something like that, and it was like, wasn't there like a problem with like, oh, we don't take that kind of paper?
No, they don't, bioRxiv didn't take white papers.
Yeah.
But didn't you mention you'd receive comments on the paper?
That was on the preprint.
Okay, so it was a different archive that you sent it to. So you did get comments on the preprint.
Right.
Correct. Yeah.
Yeah, but maybe less visible.
Yeah.
I think it was a good thing
they
to
were
talk
just
about.
emails. I just got them as emails.
Okay.
But
Yeah.
yes, yeah, this is the kind of paper. It would be nice to have an open comment section.
Yeah. Yeah, well those are great points, Doreen, and yeah, we can continue figuring out, what the best way is, GitHub
Monica,
repo, comments on the website, etc.
Monica, do you know, does NAL have anything like that they could host?
Where the requirements are sort of a forum setup.
Right, because you can think about almost all these papers are coming out for standards of recommendation for agricultural species. Maybe there might be something there. I'm not as familiar. I know that you're a little bit more familiar than I am with some of those things, but that could be something to look at as well.
There's no forum software capability to my knowledge, but I can ask and see, if we could pitch that sort of a thing that would be easier to set up. Yeah, the only communications setup we have right now is just a unidirectional mailing list type thing that's through USDA. So, but yeah, no, it's a good point to think outside the box there. Great discussion. We're at the top of the hour. I know I have another meeting. I'm sure lots of other people do. So thanks again to Adam and David for presenting and this entire working group for all the work that you've done. I think it's been a really great product for AgBioData and something that we can be proud to showcase to NSF. Thank you for volunteering your time on this and think about Lenore's comment about the creativity extension and some of the funds that we have available now for continued work. And look for those emails too, with the asterisk, of course, the uncertainty of what's going right now with funding and grants, et cetera. So, but appreciate all the dialogue, and hopefully that's not the last discussion of these non-standards recommendations. So, all right, take care, everyone. Bye.